cord because this is actually good stuff. So the ABO blood typing is really important for people to realize that's just the major blood typing. There's minor, what's called minor compatibilities too, that have antigens to big D, little d, big E, little e, and a lot of the other alphabets, if you will. So that's called minor compatibility testing. It's referred to as minor because it typically doesn't give as devastating as immune response to it, but it doesn't mean it can't be deadly. So in a perfect world scenario, every patient who needs blood, and I don't know if I really told you all this, so you should know this. In a perfect world scenario, you have time to get a sample of blood from the patient who's going to need it. And you have little samples from all your units that you have. They have, I don't know if y'all have ever seen them, but they're little plastic tubes that are crimped. And it's usually about 15, 16 per unit. So that's been donated. So what the blood bank does is take the patient who needs blood and they take their sample, their packed cells and their plasma, and then they take a little piece from the donors and they literally at 37 degrees see what they're going to be doing under a microscope when they've been mixed. So they're not just getting the blood type, major blood type correct, but they're actually also looking at what would happen in the person's body. So that's what's done for every unit that's given, except for when it's an emergency unit. And when it's an emergency unit, then you don't have time. So that's when you sometimes will give O-negative packed cells to somebody, or you might give AB positive plasma to somebody before you had time to do that. But it's rare that you all would ever give blood to a patient that didn't have that to happen in the immunology blood banking lab. The problem comes in is that as wonderful as that is, and as sensitive as that is, you can still miss some incompatibilities, especially in like the minor blood typing groups. So, that we learned ABO, blood typing major compatibility things is usually what's causing most of the trouble. But there's a whole nother part to it too. And so that's why in the blood typing lab, they're doing it all. They're looking at it all. So do errors still happen? Yeah, they do. They still happen. Yeah.
So on the donated blood, is that what you're talking about with the little samples? Is that what's folded up and like?
Yeah, they're in rubber bands.
Like a straw and rubber.
Yeah, okay. It's like a straw. So have y'all seen this?
I saw it yesterday. I went shadowing yesterday.
Okay, good. So that's exactly. So how many of you said you've given a unit? Okay, or maybe had pheresis. So when you give a unit, I told you they don't keep that whole, do they? They're going to end up kind of separating it out and doing packed cells, plasma. They're going to take the Buffy coat off and kill your white cells because your white cells can't go to anybody else. And they're going to take your platelets, put them in another unit. So they've done that, but they will also have, and it does look like a little clear straw. They let the blood, especially packed cells, run into it, and then they clamp it off. So there's a lot of little samples of it, and they fold them up. So when you pull that, if you're in the blood typing lab and you pull that unit you want to give to this patient, you're going to take that patient's sample, like I said, and you're going to take one of those little strips from that unit, and you're going to cross match it right then and see how it's looking. And they're not just looking like on a card the way we were with our eyes. They're actually keeping all the blood at 37 degrees and being very careful with it. And they're looking at it under a microscope to see if there's any reaction at all. Even with all that, though, can things go wrong? Yeah. So when they say somebody's been cross-matched and that unit's acceptable for that, that's what happened. They did that. Now, if you got somebody in your ER bleeding out, then you don't have time to do that. You're still going to do that, but you're going to be doing it while they're getting the blood. And if you were to see a reaction, you'd say, stop. Pick up the phone and say, stop with that unit. But anyway, yeah.
Now you said look for a reaction. What will you actually see? Going on?
Oh, so you have these little vials that are kept at 37 degrees. You're going to see this. You got these little vials kept at 37 degrees, and you're going to take after you've mixed the donor with the recipient, you're going to let it sit for a prescribed time, and then you're going to take that vial under a microscope, and you're going to be checking, twirling it because it's blood. It looks like blood, and you're going to be looking for any cells that might be clumpy. Because if they're clumpy, even microscopically, can you use it? No. All right, I want to look up that null thing and see what that is. Sounds like that person just can't be given anything because they are going to respond to everything. And there really are a lot more antigens than A and B and D. There's a whole bunch more of them than those. So, all right. Anyway, okay. Luckily, though, there aren't that many people that are like that. All right.
So when somebody gets a transplant, are they looking at the blood type and
That's a good
what else are they looking
Okay,
at?
so when somebody gets a transplant, they have to match the blood typing. And that's an easy thing to do, really. I know I made it sound like that's complicated. You can get a cross match done in minutes if somebody knows what they're doing. Excuse me for that. That's like so loud. Let me just turn that off right here. Billy, I don't know how to use my phone. No. All right. Oh my God, really? I don't want this phone call. It's probably a telemarketer. And I am going to turn it off. I had to do that because of this trip we're going on. Things. Okay. This conference I'm going to 12 times, but people trying to make less information. What was I saying? Transplant. Oh, so the ABO blood typing is not something that takes a long time. You can usually do that, that's like the easiest part of a transplant. But you do have to do that first. What can take a little bit longer, though, are what are called the major histocompatibility proteins, which are going to be, I told you that each individual has self-identity proteins, that little signs that are unique to them. And there's several of them. So that are unique to you. Nobody, Julie, has exactly like yours unless you have an identical twin. And most of us don't have an identical twin, do we? So we're all unique unless we have an identical twin. And these tissue markers, they're called tissue markers because they're on our cells, and cells make up tissues. So these self-identity tissue markers, major histocompatibility proteins, that's what has to be matched. Now, is there any way to do a match like you can do for blood typing on those? No. So what they try to do is match as many as possible, and they have these standards that they go by. So there's no way to match completely. So is a person who's going to be getting a transplant is their immune system going to respond to that foreign self-identity markers? They are. Tissue typing. They are, so you have to suppress the person's immune system and hope that they won't reject. That's what you have to do, is hope that they won't reject. But forevermore, that person has to have their immune system suppressed. And so then that sets them up, anybody who's had a transplant, it sets them up for opportunistic diseases. And so it's attached to them too a little bit, but they're going to die without the transplant, or the life's going to be greatly, diminished quality of life if they don't get the transplant. So they make that choice. They get the transplant and go on the anti-rejection meds. A lot is being done right now with stem cell research to hopefully someday, and maybe very, very soon, make it so that we won't have to do those anti-rejection meds because they're able to change the tissue markers and or get tissues from a person's own body, grow them, even whole organs, and be able to transplant, an organ that was really essentially theirs, and so they wouldn't have that to work to contend with. But yeah, those are the tricky things. If all we had to worry about was you must do the ABO typing first. But if all we had to worry about was the ABO typing and some of these major, minor, blood typing things, if that's all we have to worry about, it would be not a big deal, but it's not. The tricky part is getting those tissue typings done close enough that you think that you're going to have a chance that the person will reject the transplant. You know, and then we talked about, I think we talked about there's a host reject, so the host can reject the transplant, but sometimes the graft will actually start a reaction in the host too, and then that's even trickier to deal with, right? So yeah, that's the tough part. I think when we stopped with, I think what we stopped with was this, but I had kind of gone forward on this, right, and jumped ahead a little bit. But we understand that in a histology slide, it's been stained. We see these nice alveolar membranes that have a lot of surface area, so air can move in. That's got oxygen in it. How much oxygen's in air? Approximately 20%. And then CO2 can move out. And this is how we would want it to happen. Now, if the membranes became thick, obviously it can't happen as quickly as it needs to happen. We call this pneumonia. This is an acute illness, not chronic. What does COPD stand for?
Chronic Pulmonary disease.
Why is that still on there? Did I say that? That's like crazy right there. Chronic obstructive pulmonary disease is absolutely right. And so, so good. And we said there's two major causes for chronic obstructive pulmonary disease, and they're considering adding another. What did we say the two major causes are? Asthma and emphysema, and they're considering adding obesity. And so, so we get that, and Lord have mercy, I really jumped ahead, didn't I? Okay, so we said that this is chronically you're not able to move the air. So chronically over time, if you're not able to move the air, this can compromise the alveoli themselves, like it does in emphysema, or can set you up for inflammation a lot of the time. So we said bronchitis actually goes along with any type of COPD, doesn't it? Bronchitis is usually a symptom of, which is inflammation of the bronchi, which is usually a symptom of any of the chronic obstructive pulmonary diseases. We said we rarely want to treat it with antibiotics, though, do we? We rarely want to do that because it's usually not necessary to treat with antibiotics. And what happens is if you keep that person on antibiotics all the time, they build resistance. And then when they really do have something they need the antibiotics for, it might be incredibly difficult to treat and sometimes impossible to treat. So, it becomes important that we understand that now, that we need to have judicious use of antibiotics and not just throwing them out there. Now, I don't know how far to go back, but had I gotten to these little alveolar sacs? Okay, so we had talked about this, and understanding that this is where the gas exchange occurs at the alveolar level here. The rest of these structures are for moving air and conducting air, and that's an important function of those structures. Because if air gets blocked, you have 4 to 6 minutes, right? And then you're done. So we've got to have that happening. But this also incredibly important. So blood supply coming from the right pump, bringing deoxygenated blood. And as the blood is moving through, oxygen will load up, CO2 will unload, and blood will continue through the pulmonary veins to the left side of the heart to be pumped through the system. So that should be kind of familiar to you. Did I talk about great alveolar cells? I don't think I did that yet, did I? Okay, so I want you to know if we took one of these little alveoli and we sliced it, or let's say we sliced it this one because you can see the capillaries on this one, and we sliced it, we do see these alveoli are single cell layer thick. And the capillaries are how many cell layers thick? Just single cell layer thick. And they're so small that red cells have to go through them single file. So we understand that this is actually wonderful because it means that gases can go through quickly. They're only going through, one cell layer here, one cell layer here, a fluid interface here, and gases can move rather quickly across. And they're going to move by passive diffusion, takes no energy, just moving down their gradients. A lot of oxygen here, not as much here, so it moves down its gradient. A lot of CO2 here, not as much here, so it moves down its gradient. You see what I mean? And this is kind of a beautiful thing. Now, I want you to notice there is a squamous, or squamous, some people say, squamous alveolar cell. That is the cells that make up these alveoli. But there are a couple of other cells that we need to know about. These alveolar macrophages, what do you think their function is? What'd you say? Yes, you're just sort of clearing up things and eating up things. So that we know a macrophage is a big eater. They're derived from white blood cells, aren't they? And so we know in our system we have on site these policemen that are keeping things kind of in order and clear. And so this is going to be important that they're here. Could there be a problem with somebody's respiratory system if they don't have enough of these? Yes, yes. But I want you to pay attention especially to these great alveolar cells. Great alveolar cells are secreting, and you'll be asked this, they're secreting a product called surfactant. And this surfactant fluid acts like, don't say it is, but it acts like a detergent. And when you say that it acts like a detergent, your first thought is that, when you hear that, is that it cleans. That has nothing to do with what it's doing. Do y'all know how your Dawn detergent works in your sink? Do you think it's cleaning anything? It's really not. It's actually breaking bonds to help you loosen stuff, right? Including, so it helps to loosen instead of what you think of as like cleaning. Actually, if there's too much of it there, dirt start to stick to your stuff. You can have too much of detergent. But anyway, that's just, we're back to the lungs. But what this surfactant does is acts like a detergent by breaking the hydrogen bonds holding water molecules together. Now, let me say this. If y'all have ever tried to put on a wet bathing suit, was it easy? Now, I hope you weren't in a hurry. You know, weren't any sirens involved, because that becomes impossible at that time. So, no, whatever. So, anyway, it's hard. Because if there's enough water, y'all know, like even with plates, they'll stick together, won't they? They stick together if they're a little wet. It'll stick together. And that's because hydrogen bonds are holding water molecules together. But if you put a little detergent, it breaks. And so this is why when you're a fetus, your alveoli are closed. They're closed because you're in water. And as a fetus, you don't want water filling up your alveoli. Your alveoli are all closed. But as soon as you take, as a neonate, you take that first breath, those alveoli need to start doing what? Popping open. And the only way they can pop open is if there's enough surfactant. So this can be measured, and this is why y'all hear about the 32-week gestation being kind of a golden week. And the reason that you do is that is where you want to get to. Because what we see is that typically, you know, neonates born after 32 weeks or after have a much better chance of surviving and not having respiratory distress of the newborn because they have enough surfactant. Surfactant can actually be given to them, but it won't work the same as their own little gray alveolar cells. So that 32-week is a really nice week for them to get to. I've seen baby boys born, at 36, 37, 38 weeks, weighing 8 pounds, and they don't have enough surfactant, and they don't make it. It's tragic. So every day in utero is an important day. 40 weeks is full gestation, not 39 point whatever, 6 days. 40 weeks are full gestation. People should get to it. Every day is an important day in gestation, and it's mostly because of the surfactant and making sure that they have enough. If somebody goes into labor early, like at the 28th, 29th week, they can test to see how much surfactant by taking some amniotic fluid. They can test to see how much surfactant the baby has, their fetus has. And if there's not enough, they try to hold off. They try to hold off delivery. If they think there's enough, and if they think they're going to be able to support, then sometimes they have to do the delivery just to save the mother's life, right? So there's all kinds of tricky things that are going on sometimes. luckily most of the time they work out really well. But that's where you heard of that 32-week thing. Obviously 36 would be much better, but anyway, 40 weeks is full gestation. So, you good? Any questions about that? What does surfactant do?
Brings hydrogen on.
Which opens the alveoli, makes them pop open, right? And so now air can move, right? And so that's great. The pleura and pleural fluid. The pleural fluid, we talked about pericardial fluid. Y'all remember that surrounding the heart? And the pericardium is the sac the heart's sitting in. And then you have pericardial fluid and the heart's moving and changing shape. Well, pleural fluid, lung fluid, works the same. It works kind of the same. So the lungs are having to change shape and size, aren't they? Can you kill somebody just by making it so they can't move their thoracic cavity? You can't. This is, kind of tragic, but this can happen. So bear hugging somebody can kill them because they can't move. They can't change the shape of their lungs. The shape of your lungs has to be changing for air to move. And when that happens, this pleural fluid is going to reduce the friction. It's also going to create a pressure gradient so that the lungs can stay inflated. And it compartmentalizes the lungs. When you hear somebody having pneumonia, your first thought isn't, oh my God, they're going to have pericarditis. Is it? No, because typically it stays in the lungs. Now, sometimes they can become septic, which means it goes to the bloodstream. But, the lungs really are compartmentalized, just like the heart is. So it has a lot of different functions. I don't care that you know these little chemistry people, man, most of the stuff is real common sense. But again, I want to bring your attention to this. Why are you breathing at the rate you are breathing? What did you tell me is the reason? To control your pH, absolutely, exactly. But why, this is a different question. Why is air moving in and out of your lungs? Why is air moving in and out of your lungs? Are you sitting there going, well, you probably are, if you have emphysema, but or some sort of lung disease, but are you doing that? Are you doing that? You're not having to do that, are you? You're not having to do that. So why is air moving in and out? Why? And I really want you to know the reason that air is moving in and out is because as the lungs change shape, the diaphragm contracts and the lungs get larger. They get pulled down, get what makes me stand up straighter when I'm thinking about breathing. I should think about breathing more often, especially during UVA games. But anyway, I realized last night I was holding my breath for an entire segment, because I was like, I'm getting lightheaded, I'm not breathing. Okay, so anyway, as the lungs get bigger, the pressure inside the lungs gets lower than the atmospheric pressure. It's hard for us to think that the atmosphere has pressure on it, but y'all have heard of like sea level is 1 atmospheric pressure, right? It's hard for us to think that because we're not feeling squished, but actually there is pressure on our atmosphere. And it can be measured in millimeters of mercury, right? It can be measured in millimeters of mercury. So what happens is as the lungs get larger, the pressure inside the lungs is less than the atmosphere pressure. So air just gently moves in. And then as the diaphragm goes back to its original shape, the lungs get pushed, squeezed, whatever, they get pushed. And now the pressure is higher in the lungs. The intrapleural pressure is higher than the atmospheric pressure, so air moves out. So your breathing in and out is simply because your lungs are changing shape. And with that changing shape and size, there's a difference between the intrapleural pressure and the atmospheric pressure. So air just simply moves in and out. The beauty of that is that it takes almost no energy. For you to breathe in, a quiet breath in, we said it's about half a liter in, I don't know what it says, but for you to breathe in, it's going to just honestly take the muscles involved, or it's going to take a little bit, because the diaphragm contracts, and if you're having any struggle to breathe in, you're usually going to use these scalenes, the sternocleidomastoid, and even some of these muscles that we think of with the chest. So this is why when you see somebody struggling to breathe, these muscles, their head's going to be tilted back, these neck muscles are going to be straining. You're going to be seeing them really struggling. But in a normal person, you don't, do you? So it takes very little energy for air to move in. Very little, as long as everything's good. And as far as breathing out, it takes no energy to breathe out. It's called passive expiration because the diaphragm just relaxes back, and air, because the lungs get smaller, air just moves out. Fair enough? Now, when someone's sick, or have respiratory disease, or whatever respiratory compromise, then it's going to change. It's going to take calories for people breathing again, and so then it's going to change completely. Any questions for me about that? All right, so we know why, which is a different question than why you're breathing at the rate you are, but we know why air is moving in and out of the lungs. We know why, because the lungs are changing shape and the difference in those pressures. We know that we can force air. We can, if we have to, try to, breathe deeper, and we can also force air out, like if you're blowing out your birthday candles, right? Anyway, so we can do that, and we talk about pursed-lip breathing. We talked about that before. Now, I want to talk to you about when the lung collapses. When lungs collapse, it's usually partial collapse. It's a whole lung collapsing. People are, well, anyway, all of it's bad. But there's two terms that are used, and sometimes they're used, I think, a little weirdly, and I'm not sure even your textbook does a good job of explaining it. But when lungs collapse, probably one of the more common reasons you see them collapse is something called a pneumothorax. A pneumothorax. So a pneumothorax is going to be a collapsed lung due to air entering into that pleural cavity, so you lose that pressure gradient, so that lung collapses. A pneumothorax can happen spontaneously for no reason. And this is usually, if somebody has a spontaneous pneumothorax, which is kind of frightening, but if they do, they're going to have a much more chance of having it happen again. And a lot of times they don't know why they happen in some people. I had my first roommate in college, Ron, was her name. Well, she had spontaneous pneumothorax, and she did not bother to tell me that. And one happened when we were together one time. I didn't know why she passed out. I knew later that her lung had collapsed, because when that happens, they have to put a chest tube in, they have to put it in, right? So they go between ribs, wherever the lungs collapse. They go between ribs, stick a chest tube in the drain, try to recreate that pressure gradient so the lung will reinflate. And it can take a little while before it happens and stabilizes. And then when they've got that, they'll yank that tube out. Not a fun procedure to have done, I can imagine. But anyway, that's pneumothorax. Pneumothorax can also happen because of trauma. So you'll see people who have like automobile accidents that have severe trauma, contusions to the chest that will cause the lung to collapse, or they will have a breach, through, you'll see gunshot wounds, stab victims, like I said, trauma from automobile accidents. You can also see people who've fallen off ladders and stuff, and they'll think, oh, I bruised my ribs, and that's all it is. This is just a little bruised ribs. But then over the next few days, they start feeling weaker and weaker, and they just can't catch their breath. And guess what has happened? Part of that lung has collapsed, and they've got to get that reinflated. So that's a pneumothorax, and it can happen for all those reasons I just said. Then there's a very different term that we use. It's called atelectasis. It does mean a collapsed lung. Atelectasis is a collapsed lung, but it's due to an obstruction of air getting to that area. So air stops. Did I tell you most of the structures we learned about were for moving air? Well, if air gets stopped at a certain point, then no air is getting past that conductive thing, and that part that should have been getting air can collapse. Atelectasis is a term that you usually use when somebody's got a tumor, or that they've got, maybe, it's not always a tumor, so don't panic when you hear that somebody has an atelectasis. It can be a inflammation in that area, so lymph nodes can swell enough that it collapses the lung past that point. So it can be for other reasons, but it's usually an obstruction. And usually they always need to find out why. And figure that out. So do you see the distinction between those two? All right. I think I have told you everything I wanted to tell you up to this point. Yeah, okay. So we're good? Everybody's understanding what I'm saying? I'm not sounding like I'm...
How did they close the hole after they put the chest tube in?
Just switch it. Right. Yeah. So that's usually what they're doing. I haven't seen one done recently. Has anybody seen one done recently? Or maybe something else that,
Is that the type of collapse where people are in shows and stuff and are stabbing them in the chest with the needle or the syringe?
like
Compression.
an emergency one where they put a straw through. That's what that is. That and they will have to cut between two ribs and shove a hollow tube in there. So on these shows and stuff, I see them doing crazy stuff with a hollow tube. But
I've seen them where they've just taken it, like, I've been watching House again lately, and so that's where I've seen that recently. It's just like, oh my gosh, that lung is collapsed. Let's take a syringe with a needle and go cut right there.
And try to remove fluid. And try to remove, that's what they're doing. They're pulling fluid off so it can re-deflate.
Okay.
So, because what will happen is fluids will build up around too sometimes, and that will do it. So, yeah. Now, we have other reasons that we breathe as well. We breathe, I had told y'all about respiratory distress of the newborn, RDN, right? Respiratory distress of the newborn, because there's not enough surfactant, right? RDN, respiratory distress. Now we have non-respiratory reasons for breathing as well. Speaking, right? So just speaking, coughing, sneezing, yawning, those kinds of things. So we have other reasons for moving air. But obviously moving air would be a really important one. The Valsalva maneuver is when you take a really deep breath and you hold it, because now your thoracic cavity is really much larger than typical, and you compress your abdominal muscles to, and you should not, it says feces and urine. If you have to do that for urinating or defecating, there's probably some other issues you need to address it. But definitely for childbirth. So, those of us who've done that know what that can be like. But anyway, so these are all different reasons other than breathing that help us. The spirometer. Y'all haven't done that yet. Checked your pulmonary function. I want y'all to know the pulmonary function test. Is there any kind of going into respiratory therapy in here? I had one in the other class. Okay. So pulmonary function tests, there are a lot of different tests that respiratory people are going to be testing. There's a lot of different things they look at. This is FYI for you guys right here, but if you're going into respiratory therapy, I would think it would be a really good idea to kind of get this down now instead of waiting on it. But anyway, the only people that are having pulmonary function test ordered is somebody gonna ask Caitlin to have pulmonary function test done? Only if she has a doctor that, she needs to be a doctor. But, because that's just overkill, isn't it? Because no, the people who are having those tests ordered are people who have respiratory disease or compromised in some way. So today, when you all do one of the pulmonary function tests using a spirometer, and this is a device that is often used in pulmonary function tests. It's called a spirometer. Y'all should know that. Y'all are going to be doing one little test to see how much air you actually have in your lungs that can be measured as you are exhaling completely. So that's one test, right? And I see students who are perfectly healthy still get anxious about doing this test. So now imagine somebody, you remember I talked to you about asthmatics that breathing through a milk straw and emphysema, last stage emphysema, what do they feel like they're breathing through? Coffee syrup, right? So imagine taking somebody who already passes out several times a day because they're panicked about not having enough air. They have this, and you're going to tell them, I want you to do these tests where you're breathing out all your air. Or, I'm seeing how much air is moving. They're panicked to begin with, right? So sometimes they're moody if they're going to have that done. And what you completely understand that, they're afraid. And fear can cause people to act in very different kinds of ways, right? And I've seen people get judged because patients get judged because they're just not very nice. Well, guess what? We're not at our best when we can't breathe, are we? Or whatever. So it's completely understandable. I have definitely seen a lot of patients who end up going through a pulmonary function test that it's just a hard thing for them to do. So being especially understanding of those days that this is going to happen for them. But anyway, a spirometer is an instrument that's often used to do some of these tests. Y'all will do one of them today in lab. Anyway, all right, now, also, I want you to know, I want to bring your attention to it. I know this is open book, but you guys need to own this language, okay? So this is some of the terminology for ventilation. I don't have to read it to you, but y'all know some of these prefixes. If you had heard apnea, A in front of a word would mean what? It's without, so a temporary stopping of breathing, isn't it? And then if you heard tachypnea, what would you think? Fast breathing. Or if you heard eupnea, EU always means true, right? So true or normal breathing. So these are just terms that you will often hear, and you all would know what those terms mean. Cushional respiration is something we often see in people with diabetes mellitus, because they have they're usually burning fats for energy, which is then caused ketoacidosis. And because they're in acidosis, to correct for it, you'll see them doing that. They'll be doing that in their sleep. Has anybody ever seen that? So that is referred to as cushional respiration. You'll see that. Sometimes you'll actually, if a patient is you think they're in acidosis and they're actually exhibiting some signs of neurological signs of it, you might ask them to do it if they're not even in it. So I want you for a few minutes to take some really rapid deep breathing. Do that for me while I'm talking to you, while I'm doing this. So, and then, okay, that's enough. Because it can kind of help. All right. So, this is a lot of this is related to just respiratory compliance. Now, when you think about compliance, this is referring to how well you can breathe. Now, as we age, our muscles rebounding, because I said muscles have elasticity, don't they? They have elastin. It's part of their makeup. But elastic isn't about stretching, is it? What's it about? Rebound. So as over every breath, think about every breath you've taken, and the diaphragm's a major muscle, if there are other muscles involved. But it's the major muscle. But as we age, does it rebound as well? So it's losing a little of that, isn't it? So does that affect, as we age, does that affect compliance? Sure it does. And so, don't age much, you have a sense of humor. So anyway, no, but there are ways that you can actually help with that. Does exercise help? Sure it does. Exercise helps. What we know about is that larger people have larger thoracic cavities. And so we understand that too. And then we talked about obstructive disorders are really kind of anything that can block airflow. So anyway, make air be a little bit less easy to move. They're going to be, what else? Oh, and I definitely will ask this when we think about, because we say we don't have to think about breathing. Actually, when we do think about breathing, do we become a little short of breath, right? So we talked about that, but we don't. It's part of the autonomic nervous system. And what I'd like you to see here is when we do this mid-sagittal section of the brain, which we've learned, we said that this area, the medulla and the pons, are called the brain stem. And this is where respiratory and cardiac control is. So this is why we know when somebody has a brain stem injury, do they usually make it? Yeah, it's going to be tricky, and they probably are going to have to be on a ventilator, right? And it's because when we look at this medulla and the pons here, we can see these centers that control breathing in and breathing out, but also this whole, control of this. And so this is why when you have damage to brain stem, a lot of times people are on a bed. All right, so anyway, there's some people who have damage to these centers, either because of pathology like tumors or injury, that they have lost that. And now they have to voluntarily control their breathing. That means that if they're not awake and thinking about breathing, every breath in and out, they would stop breathing. So what you'll see with this are people who've had like C1, C2 fractures that have had damage to this area. and they're on a vent, they're paralyzed, they're on a vent, and sometimes they can come off the vent for short periods of time. But when they come off, they're obviously having to voluntarily control their breathing and then go back on the vent when they're not. Y'all have an insight that's talks about Ondine's curse. This Ondine's curse, this is a mythology story that they get this from. But it means that you have to mandatorily, voluntarily control every inspiration and expiration. You can imagine that there are not many people that are having to do that, because usually they don't make it, right? Or they're on the vent all full time. I had talked to you about what air is made of, approximately 20% oxygen, mostly nitrogen gas, and then the 1+% is going to be all the other stuff, CO2, water, pollutants, you know, whatever, so all the other stuff. I like this visual. Some of you don't appreciate visuals the way I do. We're all different learners. But if we thought about this being an alveolar sac, a little alveoli, and this being the blood capillary that's running by it, if we had a lot of O2 had come into that alveoli because you just took a breath in, the O2, and it doesn't take it long, will diffuse across until it's balanced. Until there's a balance between them. Just passive diffusion, no energy required. And at the same time, these are little gray solids called CO2. There's a lot of CO2 being brought because the CO2 had been picked up at your tissues, right, from aerobic respiration. And so CO2 is going to unload. So what this visual shows you is oxygen loading and CO2 unloading. And when we think about this, it doesn't take very long. Look at this, not long at all. And of course, if we're exercising, it has to happen fast, or it doesn't. So you know, hopefully this kind of makes sense. Doesn't take too long at all. I would want you to just remind you of carbon monoxide poisoning that I told you about it. Y'all know, you've heard about carbon monoxide, CO, right? CO, carbon monoxide. You've heard about that being a poisonous gas. Why is it poisonous? Why is it a poisonous, deadly gas? What'd you say?
There's no smell to it, is it? So you don't know.
Yeah, but that doesn't make it poisonous, right? But you're right, that makes it scarier because we don't know, because we can't detect it. But it doesn't make it poisonous. So it is odorless. It's colorless. Being color. I had somebody one time on a quiz write that it was dangerous because it was odorless and colorless. That doesn't make it dangerous. That is describing it, but that doesn't make it dangerous, does it? But why is it dangerous? Why is carbon monoxide dangerous?
Is it taking the oxygen away from you?
So if you had carbon monoxide, do y'all know, let me say this, do you know the little oxygen, O2, do you know what part of the red blood cell that O2 gets transported by? What's in a red blood cell that has a high affinity for O2? What's inside of a red blood cell? Would you say? Hemoglobin, a hemoglobin molecule. And one hemoglobin molecule has these 4 little iron atoms, and iron has a high affinity for oxygen. So I want you to think about it as sort of like a train with cars going through a station. So the train station is the lung, the alveoli. And as that train with cars are going through, the cars have a really high affinity for O2. So all the little O2 are jumping right on cars, right? They're getting right on. Fancy, right? And then when they get to the tissues, they'll jump right off because it has the iron has a really high affinity in the hemoglobin for oxygen. And so oxygen really likes it, but it will readily jump on and off. Are you with me? Carbon monoxide. Come up here, Aaron. Aaron doesn't mind playing this game. Y'all will never forget it. Aaron, there's our little car to get on. There's our little jump. Your oxygen. Jump on. Jump off when you get to the tissue. She does that. Jump on, jump off. Now, we're at the station. We're in the alveoli, and I'm carbon monoxide. Go to jump on. I'm carbon monoxide, and I'm sitting here, and when I get to the tissues, I'm not getting off. And when I get back to, did y'all tell me that the little red blood cell got back to the lungs? I'm still sitting here. You still can't get on. And then I'm going back through the system and I'm still sitting here. You get what I mean? And oxygen can't get on. Thank you for playing oxygen. So do you get what I'm saying? Carbon monoxide has a much higher affinity for the iron hemoglobin than the oxygen, and it won't get off. So that means your heart is pumping around blood that didn't pick up oxygen to your tissues, and your tissues are starting to suffer, aren't they? Because what's happening to oxygen levels in the tissues? And which tissues are the most affected by hypoxia? Which organ system? The nervous system. And your heart is pumping harder, trying to send blood around, but carbon monoxide is sitting on the hemoglobin, and oxygen can't get on. You get it? Does it take you long to die with carbon monoxide poisoning? Are we talking minutes? Depending on the concentration of carbon monoxide in the atmosphere, it doesn't take very long. Smokers that smoke up to just a couple of cigarettes a day, a couple, have about 10% of their hemoglobin occupied with carbon monoxide. So why do smokers, if they don't die from lung cancer, they die from what? Heart disease, because that's their hearts had to work harder. Because at any given time, 10 to 15% of their red blood cells had what sitting in them? Carbon monoxide. Who needs a cigarette? I did that the other day. It's not funny. Okay, so it's really not funny. Like, it's not funny. Okay. How many of you are smokers? Nobody's admitting it. There's got to be smokers in here. Come on. Come on. Mariah's smiling. Mariah, you a smoker? No, she's like, no, I'm smiling because I'm not. So anyway, you guys, is nicotine a tough addiction? Tougher than, say, than heroin? So, yeah, it's a tough addiction. I'm not criticizing people who smoke. Do y'all think I am? I hope you're not thinking I am. I am absolutely not. It's a really, really tough addiction. But there's so many physiological things that I think if young kids knew before they picked up the habits, that might make a difference. I don't know. I think it might. Anyway, we're continuing on. I have no idea how to get rid of these. That's sad. I must have saved it on my phone. All right, did I talk to you about this? I did, didn't I? Yes. And y'all understood it completely. I promised you that was the hardest part of this chapter. But you will be asked questions on what somebody in acidosis, somebody who has too much of this, what should happen to their respirations to change it? They should go faster. Yes, they should go faster to blow that off. That's exactly right. The rest of a lot of these are just like pictures to help you visualize how this is all running through. So, and how that if pH is the most powerful thing, unless what happens? You lose oxygen. Like if you're not drowning or somebody's not suffocating you or you're not at too high of an altitude, then pH is the most powerful stimulus. Now, if any of those other things do happen, then absolutely oxygen becomes a player. Can you have too much oxygen? Yeah, you can have too much, but let's look at too low first. Too low, and like I said, oxygen's usually little effect unless you're hypoxic, and then it becomes a player. But imbalances, so hypoxia, if you just say somebody has low oxygen in the bloodstream, you really want to know why. There are a lot of different reasons why. You could have because of you can't get gas exchange, so high altitudes, drowning. What's aspiration mean? What does aspiration mean? Choking. Choking, or you've got something obstructing the airway, right? So aspiration, respiratory arrest, meaning you just stop breathing, which can happen. People can just stop breathing. Degenerative lung diseases that are chronic, and carbon monoxide poisoning. Ischemic means your circulation, you're not getting good circulation. So ischemic attacks are going to be because of that, a lack of circulation for whatever reason. Anemic means you don't have enough hemoglobin and red blood cells. Is that right? So that's what anemic meant, and we knew that Histotoxic is because of poisons. So cyanide is one of those famous ones you'll see of these movies and stuff where somebody, well actually go like it happens too, where somebody gets cyanide poisoning. Does it take long? No, doesn't take long at all. What's a sign? What can you see that would indicate somebody's not getting oxygen? Bluey. And usually it's going to be in the lips and the nares and the extremities first. This darkening, this bluing. Absolutely. And if it goes on too long, tissue's going to die. Pathological necrosis means pathological death, early death to a tissue. And the ones with the highest demands are the ones first affected, and that's the nervous system. That's the nervous system, which is really not a good thing. Now, can you have too much oxygen? Absolutely. This is why, you know, oxygen is a prescribed medication. It's a prescribed medication, but people don't see it that way. A lot of times people see oxygen as the first step into the grave. And so they prolong going on that medication for too long, because if they go on it, they can actually slow down progressions of their disease and live longer, but they will choose to not go on it until they get very, very critical. And that's really a shame, and it's a stigma associated with it that should not be there. Nobody would prolong going on insulin, would they? So the same thing applies to being prescribed oxygen, but people just don't want to do it. Yeah, how many of y'all know people who waited very long before they went on oxygen, and they probably have been encouraged to do it much sooner, but they just did not want to do it. Now, this is going to create, if you get too much, this can create free radicals. And so we said these are going to destroy enzymes. Again, nervous system is going to be the one most affected by that. Now, the hyperbaric oxygen, the full tents are not, they're usually not used anymore, and they shouldn't be, because that pure oxygen is really neurotoxic. It's going to have effect to the nervous system first. But they can use it for isolated areas, like if somebody has gangrene, they're going to have to debride and take away the dead tissue and get good circulation to the living tissue that's there. And they can actually put the lens in a hyperbaric chamber, because oxygen is toxic to the bacterium that causes gangrene. Who remembers what causes gangrene? What's the bacterium that causes gangrene? Clostridium perfringens. Clostridium perfringens is the bacterium. It's an anaerobic. Y'all should remember that. If you didn't, want to look it up. It's the anaerobic organism, and if you just put it in oxygen, oxygen kills it. So getting those tissues saturated with oxygen is a great treatment for them. So anyway, I've talked to you about asthma. I've talked to you about emphysema. I think you guys kind of know. We said that if somebody has had a poor pulmonary compliance, and see if this doesn't kind of make sense to you, then the right side of the heart is potentially going to have to overwork trying to do this. So the right side of the heart can enlarge. It can become hypertrophic. We said hypertrophy is enlarging of muscle. So this enlarging of this right side of the heart is called cor pulmonale. Probably anybody who smoked for any length of time, their right side of their heart is enlarging. So cor pulmonale, and it's because of obstruction of that pulmonary circuit. So I'm not meaning to rush, but I know we're going to take a break here in a second. So we know lung cancer has a pretty, still today, has a pretty serious prognosis, isn't it? And a lot of times cancers, if they're caught early, we have really good results. But the problem comes with lung cancer is that a lot of people don't catch it before it's already metastasized. And that's what makes lung cancer such a bad prognosis, diagnosis with a bad prognosis, is that you have less than about 7% surviving at the 5-year mark, which is kind of sad. So there are different types of lung cancer. They're not all created equal. There's adenocarcinomas. The one that you hear about when somebody is diagnosed with lung cancer and they're usually gone within 2 months, it's over. It's usually small cell or oat cell. It's called small cell or oat cell carcinoma. So that's the one that's the least common, thank goodness, but the most dangerous, because it is usually metastasized before you know that you even have it, and it moves really quickly. So when you look at a healthy lung, well, obviously this lung's been removed, so how healthy can it be? But this is what a lung would look like in a person, that because it's just been removed. This is the smoker's lung. These are your disorders of the respiratory. We've talked about many of them. If we hadn't, we would still want to look at them. Tuberculosis is an infectious disease. It's caused from a bacterium, mycobacterium tuberculosis. So it's a pretty easily infectious disease if someone has an active case. If they don't have actively producing sputum and coughing with living organisms in the sputum, it's not that contagious, believe it or not. But if they do have active sputum, then they are contagious, and this is why they put that in isolation until they have 3 negative sputum cultures, and then they can come off of isolation, right? But it is an infectious disease. It is a disease that you all know the little skin test that we do, which is an indirect serology test to see if you've been exposed to this bacterium before. And we use that test for diagnosis purposes, right? So tuberculosis and sleep apnea, I think we've all heard of that too. So anyway, this is sleep disorders. Any questions for me? Let's take a break, and then when we come back, we are, even though, whatever, we are going to start with the urinary system. I need to go but let's take a 10-minute break, okay? Y'all get up and jump around or something. All right, so before I go off of this, let me remind people. So there will be a respiratory lab labeling, right? But I think I had given you guys the structures. I don't know. No, really.
I think you said look at the PowerPoint to go over it, like we do with the veins and arteries.
Okay, so this would be a great slide to look at for your lab, but we also know this muscle is the what? And we know this cutout here for the left side is called what? Cardiac notch. Cardiac notch, right? So actually the rest of this would be a really good thing to look at. All right, fair enough? We added those 2 things. That's great. So just as a reminder, and that will be on Tuesday morning, first thing. You will not have a lecture quiz on the urinary system that we're about to start. You won't have that, okay? The reason I'm doing this is because I'd like to erase this stuff before I. Okay, never mind. I don't care. Kind of do care. Wasn't there some slides with a lot of these? Huh?
It was in the 50s.
Oh, it was. Okay.
There you go.
Oh, 60.
All right. So this one, I'm just going to delete. And this one, I'm going to get rid of these. Really? How do you get rid of these easy? Is there a way?
Probably can't because you say
I probably can. Like, how crazy is that? Okay, whatever. I'm fine. I don't care. Okay.
Just try to redo.
If I go back to that and do what? What'd you say?
Could you go back and just where the arrow is? So,
Right here and do redo?
Yeah. That will probably undo it, yeah.
Oh, that would just take away that delete thing. I'll figure that out. Y'all are not to worry about my craziness. Okay. All right. I think I turned the recording back on, didn't I?
Yes.
Okay. Okay, guys. So I don't want you all are going to be working on your take-home class, and I am going to give you a scan time sheet just so I can quickly run through on Tuesday posted grades. Soon as long as everybody's done that test on Tuesday, we'll go over it. Y'all bring it in. But that morning at 9 o'clock, you'll take the respiratory lab, just like those structures. It will be like today with the dots on the ones that you have to do. And there will not be a urinary lecture quiz. We won't have that, okay? Because I want you to just be concentrating on the other thing. Now, on Thursday, On Thursday, you probably will have a lecture quiz on anything that we talked about in the urinary system. So do take some good notes here, because you know lecture quizzes are on just what we speak about. Fair enough? So this is starting a whole new set of information for another test that will be on urinary and digestive. And then you will have a reproductive test, for reproduction, and then a final exam. So you all know that you will have significant, you've got some other grading opportunities here to help you grades and whatnot. So any questions for me about what's coming up? All right, don't keep me honest, okay? All right, so let's talk about the urinary system. We just finished with the respiratory system and understanding that the respiratory system in many ways is an excretory system, isn't it? But it's a system where we can get rid of CO2, and that means we're getting rid of hydrogen ions, and that's helping us to maintain our pH balance. And at the same time, we're being able to pull in oxygen that is absolutely needed for aerobic respiration. So, but we have this other organ system, this entire organ system, that is going to be a major player in pH balance. So we've got two organ systems that act as buffers. The respiratory system, which acts quickly, because you can change your ventilation rate rather quickly. And then we have that which takes a little bit longer, but the urinary system. And that's going to also be a major player in helping us with pH balance. And that's going to be one of its major functions. So when we think about the urinary system, we know it's excretory. We know it's a way that, we produce urine, and in urine we think that this is a way to get rid of waste, and that's really true. But what is waste? What is waste when you think about that? We know already that urine is filtered blood plasma, isn't it? It's filtered blood plasma. And we know that plasma is mostly water, and urine is mostly water. So can we actually think about water as being a waste product? And you kind of can think about that. Are you good with that? And so we understand that however much water your kidneys let go of, this is going to be a direct way that we can control blood volume. And blood volume dictates blood pressure, right, inside the vessels. So the kidneys are going to be a major player in controlling blood pressure. We already know that. What are some hormones that we know that target the kidney to help maintain correct blood pressure? Tell me some hormones that you know that do that. A and F. From where? The heart. Atrial natriuretic factor from the heart targets the kidneys. If this sounds not familiar to you, then you know where you've got to work, and you need to be working because it should be familiar. A and F from the heart is actually going to the kidneys, causing you to lose sodium. And if so you lost sodium, water's going to follow it, so blood pressure is going to go down. What's another one? ADH. ADH. I know Andrea is not the only one who's knowing these, okay? ADH, antidiuretic hormone. Where's it from? It's from the pituitary. It's actually made in the hypothalamus, stored in the pituitary, released from the pituitary valve, and it targets the kidneys, and it's going to cause you to pull more water back into the system, keep water in the system, raise blood pressure, decrease urine output, but raise blood pressure. And what's another one?
Aldosterone.
Aldosterone from aldosterone from the adrenal glands targets the kidneys so that you lose sodium. And if you're losing sodium to that filtrate, so water's going to follow it, means you're going to I'm sorry, please. You pull sodium back in, aldosterone. It's the opposite of A and F. Sorry, I apologize. Opposite of A and F, right? So you're pulling sodium back in, water follows it, raises blood pressure. Sorry. So aldosterone from the adrenal glands. And then we also know that there's something called angiotensin that actually constricts blood vessels, right? It constricts blood vessels, and it's going to help to raise blood pressure. And when it constricts blood vessels, we're going to see how that affects blood moving through the kidneys, okay? let's see if it doesn't make sense to you that if blood vessels are being constricted because of angiotensin, what do you think about the filtering rate of the kidneys? Is it going to slow down or speed up?
Slow down.
Slow down. Exactly, right? Slow down. So again, raising blood pressure. A couple of different ways it raises blood pressure. So actually, let's take a look at the anatomy of the urinary system. On this little illustration, the urinary bladder is not labeled, but I think most of y'all would have known that that was the bladder, right? Well, this would be an easy lab, wouldn't it? So we would label the what? Kidneys. And then the structure that's carrying urine from the kidneys to the bladder are ureters, right? And then this is the urinary bladder. And then this is a little tube, a hollow tube, muscular tube, that like these are hollow muscular tubes, the ureters. This is called a urethra. Right? That would be an easy lab, wouldn't it? All right, so what'd you say? You like this one? So we're gonna have one that's similar to this, and so that should be a gimme lab grade, right, for everybody, because we do see that this looks basically straightforward. Now we got to look at what all these structures do and what's happening in these structures, and do they all need to be working effectively for the urinary system to actually be doing what it needs to be doing? So yes, it does. So when we think about kidney function, we said we already knew that it's filtering blood plasma. We know that it filters blood plasma. We know that it's going to help to get rid of waste, but at the same time, it can't let the kidneys, as it's filtering waste, it can't let the good stuff go. The good stuff in blood needs to be maintained. What did y'all tell me you find in blood plasma? Antibodies. You don't want them going anywhere, right? We need them. Same. What else? Albumin, which are, other plasma proteins that we get from the liver. We can't, they shouldn't even get filtered. Proteins shouldn't even go through the filters. They should stay back. But the good stuff, how about like glucose? And how about gases? Because there's gases and that too. How about electrolytes? Sodium, potassium, calcium, right? Magnesium, bicarbonate. How about all those things? Can we let all that go? Or do we have to keep that back? Do we have to keep that back in, right? And we can't let too much water go, so the amount of water we go has to be just let go of, has to be just right. Has to be kept in homeostasis. So we're going to see how actually all of this is really happening. And we absolutely know, since urine is really filtered blood plasma, this is absolutely regulating blood volume, isn't it? And that means that it's regulating blood pressure. So what is osmolarity again? What does osmolarity mean? Knowing the percentage of solutes that are in that solution. And we said the reason that osmolarity is important, that you know the osmolarity of blood plasma versus the osmolarity of intracellular fluid versus the osmolarity of the lymph that's bathing your fluid, is that there has to be a balance in these, because that dictates how what moves between compartments. Water. Andrea, are you laughing at me? No, ma'am. Okay, but it does. It dictates how that's moving. So good. Now, we also know, maybe we know that the kidneys produce a couple hormones that we've learned about, and one of them's not on here, but we know that it secretes erythropoietin. What is erythropoietin? A hormone that does what? That provides us with red blood cells, because this hormone travels to where?
To the bone marrow.
Red bone marrow, to target the stem cells to become red blood cells. That's how that stem cell is going to know what path to take. Is because erythropoietin got to its receptors, was brought in, and that little stem cell said, okay, I got the message, I need to become a red blood cell, right? I mean, essentially that is exactly what's going on. But we said also the kidneys secrete calcitriol. Do y'all remember calcitriol? These are hormones. If you don't know your hormones well, you should by this time. Or familiar. They need to be really familiar. Calcitriol does what? from the kidneys does what? Raises blood calcium levels by targeting, as it's released, it targets the skin, and this is going to help with the production of vitamin D, isn't it? And absorption of vitamin D, and then that's going to help with the absorption of calcium. So we get that. Now, maybe it also makes sense, and I hadn't really talked about renin too much, but this is not a hormone by itself. It's actually a precursor molecule. But renin from the kidneys, and maybe we'll remember it because of renal, whatever. But renin actually is then going to act with molecules from the lungs and the liver to get us to angiotensin. And what did we say angiotensin can do? Vasoconstricts. Is that right? Vasoconstricts. And we'll talk more about that in a minute. But okay. Now, it's also a major way we control pH. So if we have, let's say we're in acidosis, did y'all say that's having a lot of hydrogen ions in our system or a little? A lot. And what's happening to the number on the pH scale if you're in acidosis? Is it lower or is it higher? It's lower. The number's lower, but that means you have a lot of hydrogen ions. So you're in acidosis now. These hydrogen ions are going to be messing up your other electrolytes, especially your potassium. So I had talked y'all through that, I think, a little bit. Just to make sure you remember that. So since the blood is being filtered, this would be a great way if you're in acidosis to let hydrogen ions go in urine and get rid of them, right? And then as you're getting rid of them through your urine, what's happening to your pH? Coming back up into the normal range. So a major player the kidneys are in helping us to regulate acid-base balance. Takes a little bit longer than changing your respiratory rate, but not a whole lot longer. The kidneys are really a great organ system for that. And then I need for y'all to know the kidneys are detoxifying. By getting rid of waste, we also, this is an area, since blood is being filtered, that we are detoxifying sometimes drugs that are legal, illegal, whatever. So a lot of them are concentrated in the kidneys. Some of them up to 800 times they're concentrated. So is this important why we pay attention to dosaging? And is it important why we pay attention to not overdosing someone? Because what can you shut down? Kidneys. Their kidneys. Right. So same thing with the liver. Some things are clear to the liver and the kidneys. And so you have to be very, very careful because at certain concentrations, they can be toxic to one or both of those organs. Now, an average person, depending on their size, assuming on their size, might have 4 liters of blood circulating around at any given time. Somebody bigger is going to have about 6 liters of blood. That's circulating around at any given time. In a day's time, 24 hours, that, let's say 5 liters, somebody has 5 liters, an average adult. The kidneys are going to filter 150 liters of blood. I want y'all to think about that, how amazing that is for a second. You've got these organs where blood is being brought in. You can't see the blood, but well, you can see this one. So you can't see them. So anyway, vessels are bringing blood to the kidneys. You only have about, let's say you're an average, so 5 liters of blood going throughout your system. But in a day's time, that 5 liters, 150 liters are being filtered through the kidneys. That's a lot of filtering happening, isn't it? What an amazing thing, really, right? So, and what amazing organs that they can handle that much and that they do handle that much. So we're going to look at that process and see how it's done. But this is also why we understand that how kidneys can shut down if you've got something in your bloodstream that is at toxic levels. Because it's they can be so concentrated in the kidneys before you get rid of it. So the kidneys are, and I don't have evidence, but the kidneys are even playing a role with the beginning of the glucose, which is what that word means, the beginning of the glucose. So in glucose homeostasis. So when we came in here in AMP2, we understood that the pancreas was our major organ for glucose homeostasis, didn't we? But now we've seen so many other things that can affect glucose homeostasis. We saw that human growth hormone affects glucose homeostasis. We saw that cortisol from the adrenal glands affects glucose, right? Glucose homeostasis. Now we see even the kidneys are going to play a role in that. So it's a little bit more complicated than just knowing that the pancreas, but the pancreas is the major player in that. But it's a little bit more complicated than that. All right, so anyway, so what are waste products? When we talk about waste products, I want you all to have an idea about waste products, okay? And this is what I want you to know. First, I'm going to talk about azotemia. Azo is typically referring to proteins, but anyway, emia always means what as a suffix? What does it always mean? I don't care where you see it. Where does it always mean? At the end of a word, it means what?
Blood.
In the bloodstream. So azotemia means that you're beginning to get these nitrogenous waste products building up in the bloodstream. You're beginning to see them, and you can see them on blood work, on the chemistry test that you do. I want you all to know what BUN stands for. It stands for blood, urea, nitrogen. That's what that test stands for. And when you get a chemistry panel, you're going to see BUN, and you're going to see a range. And hopefully it's in the normal range, right? Hopefully it's in a normal range. If there's too much blood, urea, nitrogen, it means that the poisons are building up, toxins are building up in the bloodstream. Now, let me tell you how that's different from uremia. If somebody said, have y'all ever heard of uremic syndrome? No? Okay. That's referring to typically that your kidneys are failing. So, if you ever hear of uremic syndrome, it means that somebody's kidneys are not doing what they're supposed to be doing. Uremia is that you're getting tissues actually suffering and tissues potentially dying because of azotemia. So azotemia can lead to, if unchecked and unchanged, can lead to potentially what? Uremia. You understand that, right? All right, so this is how these terms are used. Now, there's signs, obviously, that the kidneys are failing. Now, let me talk to you about some of the waste products that we talked about in nitrogenous waste products. Now, urea is a normal breakdown product of protein. So what did I tell you? Our trillions of cells are factories of what? Proteins. So is it normal for proteins don't have a forever shelf life. They have, they get broken down to amino acids. We usually reuse the amino acids to build new proteins. It's all great. But they don't all do this. So there's sometimes breakdown products, especially from amino acids, that will go to this NH2 little thing here. This is going to be removed, and it ends up being converted to ammonia, NH3. But anyway, ammonia. Does it sound like it would be a good thing for ammonia to build up in the system? No. So you know what the liver does in response? The liver produces enzymes, and this got cut off, I'm sorry. But the liver produces enzymes that convert ammonia to urea. And then guess what our kidneys can filter? Urea. So, good. But if the liver's not functioning, because somebody has liver disease, hepatitis for whatever viral reason, or hepatitis for whatever reason, autoimmune disease, or hepatitis because of drug abuse, or whatever reason, somebody's liver is not functioning, might they start to build up ammonia in the system? And would that be toxic in the system? Yes. Yes. Okay. But if the liver is functioning, we convert ammonia to urea, and then can our kidneys get rid of it? Yes. But if our kidneys don't get rid of it, then is that going to be a problem in itself? Yes. Yes. All right. Now, uric acid, I want you all to know that uric acid is a normal byproduct, a waste byproduct of nucleic acids breaking down. What kind of why do we have nucleic acids breaking down? And what are some examples of nucleic acids? So nucleic acids, remember the nucleic acids that are inside of our cells that we use? How about DNA? Isn't that a nucleic acid? How about RNAs? All the different types of RNAs. Then we have these coenzymes. We have all kinds. How about ATP? That's a nucleotide, right? So we have nucleic acids, and as some of them are breaking down, you're going to get uric acid. But that's okay, because if your kidney's functioning, what can you get rid of? Uric acid. People who can't get rid of uric acid the way they're supposed to, uric acid will migrate to different areas, especially synovial fluid, and it will crystallize in synovial fluid. And if you take some synovial fluid from a joint and put a drop of it under a microscope with a cover slip side, and you see these beautiful yellow crystals that look like cut glass, guess what they feel like in the joints? Cut glass. That's what they feel like. Something. Anyway, what do we call that? Yeah, that's exactly right. So some people have a buildup of this, and that's just not a good thing if the kidneys aren't functioning. I should be telling y'all normal before I tell you alcohol. Is it normal to have urea? Is it normal to have uric acid? Is it normal to get rid of them through the kidneys? Yes.
How do they say more likely to drink higher, like concentrated of alcohol or uric acid?
Yeah. So there's again, this is kind of some of that's being thrown out. So yeah, they used to tell people who had gout that they shouldn't eat red meat, they should stay away from alcohol, they should stay away from these things. Now, they still, I think, encourage people to limit those things, but they know it's not directly related to that. But when we are drinking alcohol, is it a poison? Is alcohol a poison? It is. It's a poison. And where's it going to have to be filtered through? Well, it's actually filtered through the liver, through some enzymes in the liver first, but this still can cause stress on these systems. And so you just don't want to do that. You don't want to be causing stress in the system. They don't think the red meats and the alcohol as much. They still tell people to limit them. But one of the last things I read was that they don't really think so. And imagine all the people who are pissed off that they changed their lifestyles from that because of that. But anyway, what'd you say? I said boo-hoo, come a little more chair. Yeah, boo-hoo, right? And go figure. You should limit red meat, and you should limit drugs, and you should limit whatever. So, yeah. All right. So these are just normal. These are normal. We all have them, but we can get rid of them as long as what's working, and as long as we're not in what? Failure, right. So creatinine is another one. Creatinine phosphate metabolism, creatinine phosphate pathways are one of our buffering systems. And you know, we have a lot of muscle mass, don't we? And we know that creatine phosphate catabolism is just a normal thing that happens in our system. And one of the end products is creatinine. And one of the ways we can evaluate how the kidneys are functioning is to look at our blood plasma creatinine levels and then check how much creatinine we're getting rid of through the kidneys. So if there is this balance, and they can sometimes evaluate kidney function based on that. So kidney function tests are often using these, and that's why you see that. But anyway, these are normal products, and as long as our kidneys are working, we can get rid of them. So the kidneys, the urinary system is not the only expiratory system. We get rid of CO2, which means we're getting rid of hydrogen ions, through our respiratory. Through our skin, we get rid of water, we get rid of extra electrolytes, we get rid of some toxins. How many of you all you can smell alcohol in somebody's skin? It's not just their breath that you smell, you can actually get it. It's coming from the pores, you can see. In another respect, though, can the skin absorb things as well? So the skin's a major player. It's our largest organ. It's an excretory organ, and the digestive system certainly is as well. So we can also get rid of products that we can't absorb and that are not digestible, but we can actually get rid of excess water this way. So the digestive system, and then the urinary system, obviously. So we're going to take a look at the kidneys, and we're going to see how they function. I want you all to know that if we had looked at a coronal section of a kidney, and everybody knows what a coronal section is? You remove the front from the back, right? So you're looking at this coronal section of the kidney. If we, this cortex versus the medulla, the middle versus the cortex, you see the capsule here. If we were to take a microtome knife and slice, do a slice 1 to 2 cell layers thick, just 1 to 2 cell layers thick, through this capsule, through this cortex and into the medulla, and we just took this slice and then we looked at it, right? These are all illustrated, but it's not a map. What we would see under a microscope, and it would take a microscope to see this, is we would see these kind of repeating structures. Do you see those repeating structures there? Mm-hmm. These repeating structures are called nephrons. And I would want you to know that the nephrons are the functioning units of the kidneys. The nephrons are the functioning units of the kidneys. And I'm going to talk to you about what a nephron actually is. You ready? All right. So we see that it's these little repeating. This is a nephron, that's a nephron, that's a nephron, that's a nephron. And then these nephrons are like joining what are called collecting belts. Do y'all see that? Does everybody see that from this illustration? All right. So I want you to keep that in mind. And again, what did you say this whole little unit was? A nephron. And this one nephron will empty into a collecting duct, but there have been a lot of nephrons cut here, so you can see this one. Are you with me on this? All right, so do we understand that as blood is coming in to these nephrons, as blood is coming in through these capillaries, that these nephrons are actually webbed with blood capillaries? We get that, right? And even though we're going to be isolating and looking at the nephron unit itself, you understand that there's blood capillaries all around them. Fair enough? All right. So what I want you to think about right here with this one nephron, here's another nephron, but with this one nephron, I want you to see how blood is being brought into this little area that almost looks like a little cherry, doesn't it? Doesn't it? Don't call it a cherry. Okay. Here's what it really is. The blood vessel afferent, meaning bringing blood in. This is called the glomerulus, a glomerulus. And I want you, that's singular. Plural would be glomeruli, right? So the first filtering that happens in the kidney happens at a spot in the nephron called the glomerulus. The glomerulus. This is the glomerulus. Do y'all see how they have a line here saying that this whole thing is the what? So as blood is flowing in, I want you to really think about this glomerulus as being a filter. It is a filter. It's got these little fenestrated openings that are like pores, that as blood is coming in, and is there pressure behind the blood coming in? Of course, because blood has to keep moving, doesn't it? Doesn't it? So as this blood is coming in, it's being filtered, and everything in the blood plasma is lost to this filtrate except for the proteins. Red and white cells and platelets can't get through the filter, or shouldn't get through the filter. Proteins can't get through because proteins are too big. And then that is going out through this little efferent arteriole, efferent versus afferent. But in the filtrate, you just lost all this great stuff. Are you with me? And in this little tube that's close to the glomerulus, it's close. So what does close mean? What's the word for close? Proximal. Convoluted means what? No. Convoluted means twisted. It's convoluted, it's twisted. So what I'd like you to do is actually see, if you looked at this one, see how convoluted this is? See how convoluted, twisted this is? This is the proximal convoluted tubule. You understand that filtrate is what moves through the filter. The filtrate has everything in the plasma except for proteins. You got to get that stuff back because most of it's good. I just told you all a little while ago, nobody stopped me when I said this, that your kidneys filter 150 liters of blood a day. You don't pee 150 liters. So what must be happening? You're pulling back the good stuff. How much is a normal output of urine for an adult? A range. Yeah, so it's like 1.5 to 2.5 liters a day. That's true. And it would depend on how much you're drinking, right? But 1.5 to 2.5 liters a day. That is really true. That's how much, and y'all would want to know that that's normal urine output in a day for an average size adult. It's not dehydrated or overhydrated. That's a normal amount of urine. Well, if you've got 1.5 liters of urine that you're making, but you filtered 150 liters, it means that about 99% of that filtrate is being recaptured. It's coming, the good stuff is coming back. So do you understand that this filtrate, and we can look at this one because it's got the blood capillaries, immediately through diffusion and osmosis. Through diffusion, you're recapturing the solutes, and through osmosis, you're recapturing what? You're pulling back in what? Water. That is happening immediately. And because that first filtrate has to go through a convoluted tubule, does it give time to be pulling stuff back to the bloodstream? Are you with me? Then that filtrate, it's not urine yet, guys. It's not urine yet. Then that filtrate has to move through what's called a loop of Henle. I think they call it a nephron loop. It's this loop of Henle or nephron loop. And if you notice, it looks like this nephron loop gets kind of narrow right here. So what do you know that causes it to do, the filtrate inside? It slows down a little bit, giving more time for what? To reclaiming back to the bloodstream, right? So the good stuff. Only allowing enough water and waste to stay in the filtrate. Then it goes through a distal convoluted tubule. A distal convoluted tubule, right? And when it goes through the distal convoluted tubule, what do you know is happening? When it goes through this distal convoluted tubule, what do you know is still happening? Still getting stuff back into the bloodstream, right? And then this whole nephron unit at the distal convoluted tubule empties into a collecting duct. And what do you think's happening at the collecting duct still? Still reclaiming the good stuff back to the bloodstream, right? And hopefully by the time it is actually, gone through this process, you've reclaimed the fluid that you need, water. 99% you've reclaimed. And you've reclaimed the good stuff. The only thing that should be left in the filtrate should be the waste, the extra water that you didn't need that day, and the waste products, those nitrogenous waste products, urea, uric acid, creatinine, right? Those waste products are going to still be in the filtrate. And then eventually, let me say this, talking louder, like you'll understand me better if I'm talking louder. That makes no sense, but hopefully. So we got to the collecting duct, and now we're in something called the papillary duct region. Still a little bit is being reclaimed in this region, but then you get to what are called minor calyxes, and no more reclaiming is happening. So now what do we call that liquid that's inside that tube? Urine. We call it urine, right? And it goes to the major calyxes, to the pelvis, to the ureter, to the bladder, and then eventually out. So let me show you this again. When you look at this slice that was made, we understand that the nephrons are the functioning units of the kidneys, which means they're the filtering. That's where it's happening, but that's where what is also happening. Reclaiming of the good stuff. So that's happening into these collecting ducts, and then into this papillary duct, and minor calyxes, major calyxes, the renal pelvis. By the time it gets to these structures that are visible, are you getting any more reclaiming done? No. The filtrate is now, the liquid that we called filtrate before is now called urine, and it's going to move through these, you know, the renal pelvis is this area here, the ureter to the bladder to be stored between voids, and then eventually empty from the urethra, right? Are we good with that? Do you have any questions about that? How that's happening? Somebody had a big sigh, like, oh my God. But isn't that amazing? Are these amazing organs, right? Right? Okay, so this is a good picture to see because we said that blood comes in. Now, nephron units, the functioning units of the kidney, which includes that glomerulus, the PCT, the loop, the Henle, the DCT, and these nephron units, you have an infinite number of them? Or do you have a finite number? You have millions of them, but you have a limited number. You don't have an infinite number of them. You have a limited number of them. There are a lot of them, but anyway. Once you lose them, though, do you think they repair? No. No. So once you've lost enough of them, you know, you've lost kidney function. You're now going into renal failure, right? So please understand that our kidneys are amazing, but there's a finite number of nephrons. And once the nephrons are damaged or lost, they're done. And I want you to understand that as this filtering is happening, you can see that, you know, if blood pressure is too high on this glomerulus, and as blood pressure is the pressure's coming in too fast in here, what could it damage? Have you ever seen filters like in a lab or even your coffee filters where water or something went through too fast? What happened to the pores? They were torn, they were bent, and then what ended up going through into the filtrate that you didn't want to go through? The coffee grinds, right? And is it a good thing? No. But in this case, what could be spilling? You could be spilling proteins, you could be spilling blood cells. Should there ever be a normal finding of blood cells in the urine? No. Should there be proteins in the urine? No. Should there be glucose in the urine? No. So yeah, if you had those, could it indicate that the kidneys are suffering, some damage is happening to the kidneys? Yes. Yes. All right. One of the biggest reasons for renal failure is hypertension. What is hypertension? What is primary hypertension? That means that's your disease. And by the way, most people who have primary hypertension, the cause is unknown. It's called idiopathic hypertension. A lot of times we do think there is a familial. That's why they ask you, has there been family history of hypertension, right? Because we do know there's a hereditary component for that, but most of the reasons is just unknown. And that's your disease. You don't have any other disease. You just are hypertensive, right? But there's also something called secondary hypertension. And what does that mean?
When it's like a secondary, like,
To a different disease.
Exactly. What's one disease that we know is very, very common? Actually, World Health Organization says that we should be on alert because there's so many people being diagnosed with this almost in pandemic proportions, not just in our country, around the world. What's that disease that we know leads to hypertension? Diabetes mellitus. And so we understand that that puts people keeping chronic hypertension. And when we think of chronic hypertension, we understand that these afferent vessels, these are microscopic, really, vessels. These are tiny. But if there's too much pressure coming in to this glomerular capsule, is it going to damage the filters over time? And can you lose enough of them that you're now losing kidney function and now going into renal failure? One of the most common reasons for renal failure in people being on dialysis is diabetes mellitus. It's probably the most common reason. So that should make sense to you, does it? All right. So, okay. All right. Any questions for me so far?
I have a question.
All right.
What if, like, every day, almost every day, you're holding your joint because you can't get too aggressive.
It's just so bad for you, Charlotte, and I know, nurses and medical professionals, especially women. Women will hold urine much longer than men won't. If men have to go, I don't care what they're doing, they will go. They will go. I don't care if it's behind a tree or in a bottle or what. Have you ever noticed that? But guess what women will do? They will hold it for the longest time. And it's so awful for women to do that. It's so awful. We should be, and I'm guilty too, but we should, and I know you're saying because you literally don't have time, right? Are you talking about you?
Yeah.
Okay. Yeah, because your jobs get that way, but you have to take care of you before you can take care of anybody else. And that's really true. It makes me think about the airlines and how they talk about the oxygen. And they tell parents, put your own on. You won't be able to take care of your child. Do yours first and then take care, right? And it really is important. Let me tell you what else women do, and it really can lead to problems for women. Chronic cystitis. Do y'all know what cystitis means? What'd you say? Inflammation of a cyst. Okay, and what is this? Thank you, Kelly. What is the cyst? It's a fluid-filled sac. So when somebody says cystitis, what are they referring to? Because what is a fluid-filled sac in the urinary system? The bladder. So if you ever hear this term cystitis, it means that the inflammation is of the urinary bladder, because the bladder is a fluid-filled sac. What's the fluid inside that sac? Urine. It's a fluid-filled sac. So cystitis means that you actually have inflammation of the bladder. Y'all have heard of urinary tract infections, but that's not very specific. Some women have just urethritis. Where is their inflammation? Of the urethra. Some people have cystitis and no urethritis. And so what is cystitis again? So in a urinary tract infection, you really should be more specific and know exactly where the infection is. You can have ureteritis. Where is that inflammation of? The ureters. Or you can have nephritis. And where is that inflammation of? The nephrons, the kidneys. Right? So let me say this about women. Women have practices that holding urine can set them up for urinary tract infections. It can set them up for their bladders becoming distended and then not being able to empty completely. Women, they've done studies. We know this is female behavior, nothing that we should be proud of. We should actually realize this is silly of us. Women will sometimes urinate, but they'll only urinate until the feeling of needing to go is over, and then they get up and keep going. But they've left urine behind in the bladder. And so this is a real problem for women because it sets them up for chronic urethritis and sometimes cystitis, and that can lead to interstitial cystitis, which is a chronic type of urinary tract infection they pee. So if you've ever, the next time you pee, think of me, and when you thought you were finished, sit back down. And if you have enough that's a half a teaspoon left, you're setting yourself up. That you can still urinate, you're setting yourself up for urinary tract issues. So empty your bladders completely, frequently void, frequently take those breaks. You have to take care of you before you take care of somebody else. Being out for a week because of kidney disease, a urinary tract infection is not going to help us just because you can get them, whatever, that second. Take care of yourself first. Yeah.
So it's a weird that sometimes I sit in the toilet at home for a long time because I know I'm not done urinating.
Well, I don't think that's weird. I think a lot of people have that sensation where they think they still need to go, but they're not going. So in a perfect situation, you should be able to completely void. When we void urine, we don't feel it, or we should feel it. People say it's very painful when they do feel it. But when you relax those sphincters so that you can void, your bladder is spasming. Now it's not supposed to be spasming enough that you feel it. People who do feel it usually are in pain, like that causes pain for them. But it shouldn't take too long to completely void urine. It should not. And so if it is taking you too long, then yeah, there's something probably wrong.
What's the
But I don't know
feeling?
what's wrong. But
What's
it's
the
just,
feeling of not having finished, but you are finished? You know, you're not completely done,
You're not
but you're
completely
not.
finished,
But you have a feeling. What's
all right,
that?
well, I don't know. Uncomfortable. So I don't know. You all heard of the Kegel exercises, right?
Yeah.
Where you're supposed to help the pelvic floor muscles, can help with all of this, right? It really can. This can help. How many of you do those? I had a friend who said she remembers to do them anytime she's at a stoplight. So if the light's red, and I think about Donna a lot now. I think, okay, she's at a stoplight. I know what she's doing. She's doing Kegel exercises, right? But this can really help, and I don't know what we're talking about. What were we talking about? Oh, so yeah, you're supposed to completely void, and you're supposed to void frequently. So there's something about urinating that they talk about in urine itself. And they talk about it in C's. The C's is for urine should be, and a lot of people say, my urine is so clear, or so colorless, is there something wrong with me? No, it's supposed to be clear, colorless, and copious, which means a lot. So when you urinate, you should be urinating a lot. Because actually the act of voiding is clearing that urethral opening. Because it's normal for you to have bacteria there, and you want to keep them sort of rinsed out, and so it doesn't move up the tract, right? So now urine can also have a color, but the more color it has, the more what does it indicate? Potentially dehydration. So it's concentrated, and you're potentially dehydrated. If you're well hydrated, your urine will be clear, colorless, and copious. And the clear is really good, but the colorless is really like yellow, right? Can other things affect color of urine, like diet and medicines and all kinds of things? Yeah, so other things can affect it, but that's what it should be. It's thought that in this country, about a quarter of adults are walking around dehydrated, which is really kind of crazy because we have a lot of water available to us. But I think our lifestyles get a little busy, or we do things that, we don't hydrate. How many of you know you don't drink enough water? I know, I don't. I drink coffee, but anyway, but yeah, so we probably all could increase our fluid intake, which would maybe help us with all of that other. You guys see this right here? This will be and this will not be Tuesday. But the Thursday, when y'all do the lab for the respiratory, I'm going to want you all to do this as a lab quiz. Because is this really anatomy? It really is. What this is showing you is where the first filtrate happens was where did I say? And you can just say glomerulus right here. But where does the first filtrate happen? At the glomerulus. Then the filtrate moves into what? The proximal convoluted tubule, then it goes through that loop, then it goes through the distal convoluted tubule, then that nephron that filtrate's going into the collecting duct, which is collecting from a lot of different nephrons, isn't it? And then the papillary duct to the minor calyx. Oh, that's making me dizzy. Did y'all see that shaking? Mm-hmm. Was that just me? And then the major calyx, the pelvis, the ureter, the bladder, the urethra, and out. Right? So this is really anatomy. It's a little bit more than kidney, ureter, bladder, and urethra, but not a whole lot more. And I would hope you all would understand it by Thursday. Okay, so by Thursday. So on Thursday at 9 o'clock, y'all are going to have a few things you need to do for me. For you, not for me. It's not for me. None of this is for me. But it's for you, so that it's just an understanding of how the kidneys are actually functioning. How do you feel about that?
The collecting duct, is that where the urine starts to collect, or where would
You're still reclaiming.
Okay.
You're reclaiming a ton of water at the collecting ducts. The collecting ducts are actually where ADH targets. You know how you all learned that the hypothalamus releases ADH, targets the kidneys for you to pull back in water, and that's where the ADH is targeted. those collecting ducts. You're pulling back in a ton of water. Let me show you something that might make this a little bit like when you're seeing this, like they've straightened it out, all the tubes, they've sort of straightened them out, but you are reclaiming so much stuff. And it's not going to be urine until you stop reclaiming. Then that filtrate, we can call it urine then. Does that make sense? Not until you're like through the papillary duct.
Oh, okay.
Yes. And then you're calling it urine at that time because it's not going to be reclaimed in that minor calyx. Yes.
You said that the flow would be on Thursdays. So last week Thursday. Can you go over what all we have to do that day?
So on Thursday morning, you have the respiratory lab that you will label. Does everybody know the structures? Okay. And you will list
Thursday or Tuesday?
Thursday.
The following Thursday. I thought
Respiratory is on Tuesday.
Respiratory is on Tuesday.
That's why I was asking.
Oh God. Okay. Okay. Oh my God. Okay. I'm getting confused now. Okay. Sorry. I'm thinking about. Okay. Kelly, thank you. Thank you. Everybody. Okay. So this Thursday we're out. Y'all don't come. Everybody remember that? Right. And this Thursday you work on your take-home test. Right? Tuesday when you come in, please bring them. You must bring them at that time. Right? Okay. And Tuesday, the 16th. You have the 16th. Thank you. The 6th, Kelly, thank you. The 16th, you have the respiratory lab. And you will have that flow thing.
On Tuesday?
Yeah.
Should we have it Tuesday?
Wait, you don't want to do the respiratory one day and then this one
Yeah.
another
You
day?
initially had
I
said
initially said that's all you have. That's all you have. I haven't said that.
Yeah.
Okay, so I initially said that's all you have. Okay. Aaron's like, oh my gosh, she's lost her mind. No, I haven't. It's just, yeah, I have. Okay. But as long as y'all all agree, you'll make sure that the right thing happens, right? So yes, I had said that all you have to worry about is the respiratory for Tuesday. I had said that. So hold me to it. That's all you'll have to do. Then on Thursday, you're going to have two things. Thursday, you're going to have a urine quiz, lecture quiz, a urinary lecture quiz, which is a typical lecture quiz. And you will have this, let's count this as a lab, right? Because this is essentially what? Anatomy. So thank you for getting that straight. Is everybody good about what's coming up the next week? So on Tuesday, you're bringing in your take-home test and handing that in. Hopefully we can go over it. Everybody will be finished. Then you are doing the lab on Tuesday for respiratory, just labeling. That'll take a minute, right? Because that's not going to be bad. But on Thursday, I hope that you would spend the time reviewing the urinary lecture notes, and you will learn this filtrate flow, which is anatomy, and you would be ready for those two quizzes on Thursday. Those two quizzes on Thursday morning, the 18th. Now that I've said that out loud, and you're gonna hold me to that, right? Everybody in here, so but anyway, I'll try with number two. Okay. All right, all good stuff to remember. Okay. Right now, we're going to go just for a little bit more, and then I am going to get you guys to do the spirometer testing, and I'll show you some the respiratory structures you would want to see too. By the way, the next week that we come in, we're going to be doing digestive too. We'll probably finish the urinary. You're going to check your urine next week. Y'all are going to have the opportunity if you want to test your own urine, and then. The week after that, you're going to do rat dissections because we will have looked at the digestive system and you're going to dissect whole organ systems from the rats. There's that lab for that. But anyway, we'll be doing quite a few things in the next couple of weeks because, y'all, we're going to be almost done. right? We don't have that much more time in here. So it is going to be important that we stay on. What's this? Are we good? Yes. All right. Okay, thank you. All right, for helping with that. Now, here's where you see those little filtering pores. So I think these things can sort of help you visualize what's happening. And we had talked about the idea that there's a certain amount of pressure coming in, and that's helping filtering happening. But if the pressure's too much, would that be a problem? Yeah, so that would be a problem. And urologist, what's a urologist? Someone who specializes in the kidneys, and this urologists sometimes have to figure out like how much of the kidneys are functioning if they're worried about the kidneys not functioning. And so to do that, they try to establish what the glomerular filtration rate is, which means like how much urine is being filtered in a given day. And we said from 1 to 2 liters, usually about 1.5 liters of urine is produced a day. But we know that in an adult, it should be about 150 liters is being filtered a day. So 99% of that is being reclaimed. Now, let's see if it wouldn't make sense about this, if we could walk ourselves through this and see if this doesn't make sense. If somebody's glomerular filtration rate is increasing, would it make sense to you that more urine is being produced? Yeah, because more filtering is happening. So more urine is being produced. Is it possible, do you see that it could be possible that dehydration could occur? If that is happening, it's possible. And with that, electrolyte depletion. And what do we say our electrolytes provide for our body? Essentially what? Electricity. It's essentially keeping the millivolt charges on our membranes, allowing for resting potentials. And then because we have these resting potentials, we have action potentials that can actually be excited and cells be innervated. If you had a decrease, if for some reason you're not filtering, you have a decrease in the glomerular filtration rate, does it make sense to you that azotemia will start, which is the buildup of these waste products, these nitrogenous waste products, that could end up leading to, what did we say? Azotemia can lead to uremia, right? So it means essentially that we have to be able to control glomerular filtration rate. Our glomerular filtration rate is going up or down depending on what's going on in our lives all the time, but it should be remaining in homeostasis. We do not want it too high, and we do not want it too low, do we? And it's all part of this autonomic nervous system control. The sympathetic control is playing a role in this. So let's look at what was this little capsule called? It's probably labeled. What's this called? The glomerulus. This is glomerulus, right? This right here is the glomerulus, this little capsule. So we said we know the afferent blood vessels bringing blood too, and it's being filtered right here. You have these cells, these nerve cells that have one function on this vessel, and that's to determine the amount of stretch of this vessel. So if there's too much blood coming in, the pressure's too high, what kind of signal would be sent? Yes, to control it, right? That's what should be sent if you're keeping it. Yeah, so if everything's working the way it's supposed to be, this is the way it should be sent. Now, the beautiful thing about this is that, even though these are like little, really, look at this, 1 cell layer thick structures, even though they can withstand a significant amount of high blood pressure. Because you all know that when you're reading these systolic values, some of you had them reversed. You had the high number on the bottom, low number on the top. But I hope everybody at this point kind of understands blood pressure. Systolic blood pressure, we said, should be lower than 140, shouldn't it? Shouldn't it? It really should be lower than what? 120. But we know that blood pressure can go up in stressful times, and we know that that's going to be okay. And we actually know that it can actually even go up higher than that. But these glomeruli can't withstand it being up high for long periods of time, because then that's going to start to damage them, right? So we also understand that these glomeruli aren't really under this pressure. This pressure is going to be arterial blood pressure that's red. At these vessels, it's much lower by the time it gets here, right? So what you are seeing right here is actually arterial blood pressure, right? Does that make sense, y'all? Because y'all saw, if you go back and look at blood pressure, you'll see that at the arteries that you're measuring, usually the brachial artery, if you're measuring, then in the arteries, yeah, the pressure's really high, but as it gets to the arterioles and even toward those capillaries, the blood pressure goes down. Right? So obviously, this is looking at systolic blood pressure, but it's much lower here. But over long periods of time, people who have uncontrolled hypertension, for whatever reason, whether it's primary or secondary, these start to become damaged. And you told me you have a finite number of these. So that means that you're starting to lose kidney function. And that's just not going to be a good thing, right? So when your glomerular filtration rate's really high, it's going to be sensed by these cells, and then you're going to get constriction, and then that's going to bring the pressure back down, right? So that's going to be all controlled. You don't have to think about it. It's actually just being controlled. Now, let me show you this. I like this little slide, and this might be one of the last things we do for today. But you said your kidneys produce renin, which is actually not really a hormone. It's a pre-hormone. It's a precursor to a hormone. But it's going to act on it's going to act on products from the liver and the lungs to get you to angiotensin. And angiotensin, you said, is going to vasoconstrict. Did y'all say it's going to cause vasoconstriction? Which is going to then raise blood pressure. But let me say this, it's going to also get your hypothalamus signal to the hypothalamus so you're thirsty. And if you are thirsty, what are you going to be doing? Drinking. And if it also talks to the adrenal glands that produce what? Which is going to cause you to conserve sodium and pull water back in. And all those things are going to do what? All these things, three things together are going to do what? This should be a review of the hormones, because you already had these hormones. So that's like a review, isn't it? All right, so we'll stop right there. How do you feel about your kidneys? All right. All right, so we are going to use the spirometers to do pulmonary function.